Aloha. Hi, everyone. This is Yusuke Marikawa. Here's a study guide for the upcoming final exam for Biochemistry 441. So be aware, it's going to be on May 10. The time is important. It's starting from noon, 12, okay, from noon. In the same room, this ART 132, the big room, okay? But just be aware, starting at noon, okay, 12. And the exam's going to have 85 multiple choice questions to 70 from my lectures, the lectures I gave after exam 3. So all those lectures I gave. And so this video covers those 70 MCQs. The remaining 15 questions are from you guys, research presentations based MCQ that you submitted to your advisors. And I'll choose one from each group. So it's going to be a total of 15 questions. So pay attention to your, fellow students' presentations and understand what they study or, figured out, researched on. All right. So anyway, we're going to focus on the 70 multiple choice questions. I'm going to go over all the lectures from the first one to the last one, just like the last time I did. All right. So focus on any yellow highlighted, those sentences or names or phrase or concept, those things, you see those yellow box or yellow highlights. Those are the ones you want to study very intensely. All right. So the first one is amino acid biosynthesis, medical correlates. So we went through those hem synthesis and so on. But you need to know what is hem, the hem is made of. Okay? They're made of glycine, succinyl-CoA. Only two kinds of ingredient you need to make hem. Okay? Glycine, succinyl-CoA. And you put the succinyl-CoA, glycine, one each together, you create this compound ALA. Just remember this abbreviation, ALA. Okay? Succinyl-CoA plus glycine makes ALA. Then you put two ALA together, makes polyphobilinogen. Yeah, two ALA together to make polyphobilinogen. Here, it's important thing is this reaction is catalyzed by this enzyme, but enzyme name is okay. You don't have to know, but you need to know what is cofactor. Zinc is involved, but it's necessary for this polyphobilinogen synthesis from ALA. Okay, I'm going to come back to that zinc aspect later on. So now you put this 4 of polyphobilinogen together to complete hem productions. But you also need additional, those metal, Fe2 plus iron, yeah, to complete this hem synthesis. That's the extent you need to know about hem synthesis. And be aware, the thing I just emphasized, zinc necessity for this polyphobilinogen synthesis is one of the targets of the lead toxicity. So lead causes various toxic problems, and it affects the zinc-dependent enzyme. So it's interfering with this hem synthesis in the end, causing anemia. Right? Now, focus on this degradation of hem. So there is quite a long pathway from the beginning of the hem. First step is the macrophage. So macrophage captures those old red blood cells to start this hem degradation. You need to know the name of this products, hem, biliverdin, and bilirubin, together with each chemical's color. Okay, hem is kind of purplish, biliverdin is greenish, and the bilirubin is more orangish. Okay, so that's what happens in macrophage. Now macrophage is going to spit it out into the bloodstream, but it's bound by this albumin protein. But anyway, right now the focus is macrophage is doing this initial degradation and the changes of this hem to this bilirubin, intermediate biliverdin, and the color is green for biliverdin, orangish for bilirubin, because it's kind of related to the blue's color changes over time. Now this bilirubin picked up by the liver. Okay, liver picks up the bilirubin from the blood and change into more soluble form by its conjugation. Okay, know that liver is responsible for conversion of bilirubin into more soluble form. And here, this sugar called a glucuronic acid is attached to bilirubin. That makes it more soluble. Okay? UDP, glucuronic acid, is the substrate and transfer this glucuronic acid portion to this bilirubin. Okay? And, all right, it's going to the bile, to the intestine. Now, in intestine, this conjugated bilirubin is further converted to stercobilin by the bacteria. And what you need to know is the name of this product and the coloration, okay, brownish color for stercobilin, which is the reason why your poop is kind of brownish color, stercobilin. And some of these products going to go into the circulation, go to the liver, the kidney, and kidney is going to convert it to urobilin, which is yellowish. Okay, so that's the color of your pee. Okay. So you need to know those names of this compound, which is a product of bilirubin degradation, and the name, right? Color and name. And notice a clinical condition called jaundice, which is due to increased bilirubin in the blood. If something happens, you don't need to know all those specific causes of underlying disorders. But what you need to know, whatever the cause is, if the bilirubin end up accumulating due to various disorders, it manifests as jaundice, yellowish coloration of the skin and eyes due to increased bilirubin. And that jaundice also happens in infants in a natural way. But sometimes it persists or is very severe. So infant jaundice in a very severe case, you have to resolve it by going through this phototherapy. So you need to know what it is, phototherapy, which is going to shine in blue fluorescent light, which this high energy light, blue fluorescent light, will convert bilirubin into soluble isomer, okay? Change into soluble isomer. By doing that, it's going to be excreted more easily. Yeah, soluble, easy to excrete. All right. And let's see. I think that's the extent of this lecture. All right. So moving on to the next one, which is about obesity. What you need to know is this. Body mass index, BMI. You need to know formula. It's basically, I'm going to ask again the kind of calculation questions with a very simple numbers, easy number, 100, 200, those things. And the heights in meter is also easy numbers. So you shouldn't require calculator or anything. You should be able to calculate. So need to know it's weight divided by height square. Okay, square. Make sure you divide twice, basically by square. That's number one. And number two, what you need to know, if the BMI is higher than 30, it's obese. This is a general categorization. If you calculate your own BMI, if it's more than 30, you are classified as obese. Okay, more than 30 obese BMI, right? And this is okay, in your regular life, this kind of awareness is important. But for the final exam, that's okay. Now, we're talking about absorption of dietary fat. You're going to eat fatty stuff. Fat basically is the TAG, triacylglyceride. That's basically molecular identity of fat. Right? So you're going to absorb this fat by going through these steps. Okay? TAG in diet need to be digested by pancreatic lipases. Okay? Pancreatic lipases chop up this TAG into monoacylglycerol and free fatty acid, like this. Okay? So you need to know that. And in the intestine, those are the forms that are absorbed by the intestine, but intestinal cells are going to put those things back together into TAG. Okay? You need to know the enzyme name that involved in this, and those products that gets absorbed by intestinal cell packed into TAG. Okay? And this TAG is going to be distributed throughout the body through chylomicron that you need to know. So intestine, after absorbing TAG, you package into chylomicron to deliver to the other part of the body. Okay? Intestine makes chylomicron to distribute this TAG, right? And notice the name of this medication for diabetes is basically preventing this pancreatic lipase. Yeah, so by doing that, your intestine cannot absorb TAG because it's not split into this monoacylglycerol fatty acid. Yeah. So that all this start inhibit lipases, pancreatic lipases, to reduce absorption of lipids by the intestine. Know the name of the drug and its action and its target. And you also need to know that sugar can be converted into TAG, and some detail which focusing on this dihydroxyacetone phosphate, which is one of the glycolysis product, right? And gets converted to glycerol 3 phosphate, which is the core, yeah, this portion of the TAG. So this conversion is one of the essential or critical step of making fat out of sugar. Okay? Conversion of DHAP or dihydroxyacetone phosphate to glycerol 3 phosphate. Okay, DHAP to glycerol 3 phosphate. Okay, as a mean to make fat out of sugar. And the feeding behaviors or appetite control, and brain is involved, and know the specific region of the brain. Hypothalamus has those specific neurons that will control appetite. Okay, hypothalamus, cells in the hypothalamus, neurons in the hypothalamus controls feeding behavior, increasing appetite, decreasing appetite. Probably you don't need to know what specific those neurons or neuropeptide is involved in which, yeah. Don't worry about it. But what you need to know is next hormone, okay? Leptin. This is a hormone that's circulating throughout your body and originally secreted by fat, okay? Secreted from adipocyte. Okay, so know that leptin is secreted from adipocyte. So if you have more fat, you're gonna end up having more leptin in your body or circulation. And leptin affects the hypothalamus to decrease appetite. Okay, leptin decreases appetite. You need to know that. And the leptin is secreted from adipocyte. That's also you need to know. Okay. Next hormone that control appetite, in this case decreases appetite, is peptide YY, produced by intestine. That's the extent of information you need to know. Peptide YY produced by intestine that decreases appetite. Okay. And number 3, this one, ghrelin increases appetite. Okay, ghrelin increases appetite. So make sure those hormones I just mentioned and its function, whether it's going to decrease or increase appetite. And also, where does it come from? Those organs, tissues that hormone is derived from. So in the ghrelin, it's stomach. When your stomach makes a noise like this, and it's going to produce ghrelin, right? That's it. All right, moving on to the next one, nucleotide biosynthesis. This is a very long lecture, but I'm gonna point out what are the most important things for the exam. And just skip all this. Now, certain specific in terms of terminology or structural detail, you need to know. So the term base is this portion of nucleotide, either purine or pyrimidine, like a one-ring pyrimidine or two-ring purines. Those portion is called base, okay? When the base is attached to sugar, like a ribose or deoxyribose, those two together is called nucleoside, okay? With a sugar, nucleoside. But if this nucleoside has some phosphate, 1 or 2 or 3, doesn't matter. If you have phosphate, it becomes nucleotide. All right, make sure you can distinguish. So if you're given a structure, some image, figures showing this base sugar with a phosphate, you call it nucleotide, okay? If the image is missing phosphate completely, but has a base and sugar, that's a nucleoside, okay? If the image doesn't have sugar, just two-ring purine or one-ring pyrimidine only, that's base, okay? So know those terminology, all right? And of course, you have to know pyrimidine, purine, that's the actual, those molecules that are members of pyrimidine, cut the pie and pure as 2 gold rings. Whatever the way you prefer, just remember, okay? CUT for pyrimidine, A and G for purines. Okay. Now, making of pyrimidines. Okay, so there's some kind of summary diagram here. Those are kind of important, but I'll specifically point out which aspect is super important. The first step is making this orotate, yeah, for the pyrimidine synthesis, one ring structure, pyrimidine synthesis. Orotate is the first the portion that we're going to synthesize, and made of three things: glutamine, aspartate, and CO2. Okay, glutamine, aspartate, and CO2. Okay, know that name of these two amino acid: glutamine, aspartate, and CO2. And the first step is put the glutamine and CO2 together to make carbamoyl phosphate. So if you look at this step, this NH2, this amino group of this carbamoyl phosphate, comes from glutamine's NH2. Okay, so that's what you need to know. This portion is from glutamine, right? And this enzyme is involved is the carbamoyl phosphate synthetase 2, CPS2. And this is very similar to the one you learn through urea cycle CPS1, but there's a big differences, okay? Lots of differences between those two, even though it's performing similar biosynthetic process like making carbamoyl phosphate. But the location, the ultimate products they're gonna make, and nitrogen source and regulators, those are different. So this whole thing is, we box with this yellow things. This entire that slide is important. Just know if you can understand the purpose in urea cycle, trying to take care of ammonia, nitrogen waste, yeah. So that's why this nitrogen comes from ammonia. But for making the pyrimidine, yeah, CPS2, which is happening in cytoplasm, it's not coming from ammonia, it's coming from glutamine. That sort of thing. Yeah. Also the regulators. Okay. And let's see. Okay, so once you make the orotate and put together with ribose phosphate, and you have this OMP, orotidine monophosphate. So next step is to make this UMP, uridine monophosphate. So this is one step on reactions, decarboxylation. So simply removing this carboxylic group of OMP, you can make this UMP. Yeah. So decarboxylation OMP makes UMP. This heading is pretty much what you need to know. And now we're further converting this U into C or T. Okay. So converting U to C, we require receiving this NH2. Okay. U to C, receiving NH2 from glutamine. Okay. So remember the source of amino group of CTP or C, okay, is from glutamine, right? So that's the extent you need to know. And then U to T conversion, in this case, you need to receive the CH2, extra carbon, yeah, CH3. And that comes from THF, tetrahydrofolate. Yeah, this carbon comes from tetrahydrofolate. And this one, you also need to know the name of enzyme, thymidylate synthase. Basically, this is thymidylate, this TMP or deoxy TMP. So thymidylate synthase, okay? So that will receive one carbon, CH3, from tetrahydrofolate. Okay. And remembering the enzyme name is important because you need to understand how this cancer drug, 5-fluorouracil, okay, 5-fluorouracil function or act as a cancer drug. It's basically inhibiting this thymidylate synthase I just mentioned. Okay, so by inhibiting this, the cancer cells cannot change U to T, therefore cannot make DNA. That's it. Okay. Know that fluorouracil, very commonly used cancer drug, it inhibits thymidylate synthase. Okay. Now, moving on to purine synthesis. And yes, some parts definitely you need to know, but I will just summarize. Probably the extent of what you need to know is this thing. I will ask questions that are focusing on the two aspects of this two-ring structure synthesis, which is there are two carbons that come from the THF. Okay, don't worry about this detail, but it's some form of THF is giving one carbon here, another carbon here. So two molecules of THF are contributing to this two-ring structure. Carbons, carbon donor. Okay. Also, glutamine, two molecules of glutamine contributing two nitrogens. A nitrogen donor. So glutamine serving as nitrogen donor. That's the extent you need to know to make this two-ring structure. Okay. THF, two THF contributing two carbons. Two glutamines make giving just two nitrogens. Now you're going to change this IMP. So that's the first step of making purine change into either A or G. Okay, so you have to make a choice. So this is the kind of extent what you need to. Conversion of IMP to either AMP or GMP. Now, both pathways requires receiving this amino group. So the question is, where does this amino group come from? That's going to be the questions, okay? So for AMP, for making AMP, you receive amino group from aspartate, okay? For AMP, aspartate, okay? For GMP, glutamate, yeah? It's just going to same letter, so it's going to easy to remember. Glutamine often serves as a nitrogen donor anyway, so GMP's amino group, nitrogen, come from glutamine. AMP's nitrogen from aspartate. Okay. Yeah, so glutamine for THF for carbon. I already emphasized that earlier on. So now you're going to convert this ribonucleotide into deoxyribonucleotide. So deoxy, so it's a reducing kind of reaction. So what you know is that there's an enzyme, a single enzyme, ribonucleotide reductase. It's responsible for changing ATGC, all those bases, regardless of bases. They're changing to ribo version, to deoxyribo version. The single enzyme does that conversion, okay? And what you need to know, that reaction happens only on the diphosphate version, okay? Not the triphosphate, no monophosphate, only diphosphate, okay? A DP or C DP, all right? And GDP, there's something DP. It works on only those nucleotides. You experienced those sample questions or quiz questions already, so you should be familiar with what I'm getting at, right? And the question is going to be, could be, there's a cancer drug called hydroxyurea. This is targeting this ribonucleotide reductase. And that's the one responsible for changing this, that sort of thing, ADP, GDP, CDP, those things. Yeah. So there will be a connection here with the one that I just mentioned. And now that this is the third cancer drug, methotrexate. Okay, what is the target of methotrexate? There's an enzyme name shown here, but I want you to remember is that it's affecting, or it's inhibiting the production of THF from the dietary stuff like folic acid, vitamin B9. So methotrexate inhibits THF formation, thereby inhibiting purine formation or even thymidylate synthase I just talked about earlier. Yeah, whatever the reaction that requires THF will be affected by methotrexate. Yeah, so that yellow box, those portion, you need to know. So the folic acid, the precursor of the tetrahydrofolate, and this is an important vitamin, of course, and just need to know, as a sort of knowledge you want to have in your life, is that it's linked to the birth defect. The deficiency is linked to birth defect. So the women of reproductive age, whether it's already pregnant or not, actually should start before getting pregnant, and that daily intake of folic acid is important. Kind of solid studies show that it reduces the incidence of giving birth to this baby with a birth defect like this. So folic acid is the one the women of reproductive age should take to prevent birth defect. Okay. All right. Next is salvage pathway of nucleotides. So I emphasize there are three kinds of these enzymes that's involved in the digestion of DNA step by step. But just focus on this middle one, nucleotidases. Okay, not the nucleosidases or nucleases. Nucleotidases, which act on nucleotide. Tide is a phosphate. Yeah, you have this base sugar phosphate nucleotide. So this enzyme act on this nucleotide to chop off phosphate, that dephosphorylation, yeah, chopping off phosphate to release nucleoside. So that's nucleotidase. And focusing on the purine salvage pathway, and there are two enzymes that are changing this purine base into purine nucleotide. And one of the two enzymes are very, very important, HGPRT, hypoxanthine guanine phosphoribosyl transferase, or HGPRT, changing hypoxanthine base into IMP or guanine to GMP. Yeah, those two jobs, two reactions, HGPRT. And this enzyme is particularly important with respect to uric acid formation in our body. And okay, uric acid formation. Okay, that comes from those purines, okay, conversion of purine into uric acid. Now, there's lots of pathways here, but what you need to know is that the final step of the uric acid formation from those purines is this enzyme, xanthine oxidase. Xanthine oxidase is responsible for making uric acid. Final step, okay? Xanthine oxidase. And here, there's the foods that will increase the uric acid level you should avoid to prevent the gout. And I talked about those fructose, high fructose kind of beverages, drinks and foods and so on. That will increase the uric acid through this a little complicated mechanism. Yeah, but you don't need to know that detail. It's kind of cool, but probably I'm not going to ask that detail. But what you need to know, it is fructose that's going to increase the uric acid. Okay, glucose is not going to do anything like that because glucose goes through the glycolysis. It has to be regulated. That's fine. But the fructose unregulated, that's the reason why it ends up with the uric acid. Okay, fructose increases uric acid. Right. So the reason why I emphasize xanthine oxidase is it's a target of this common medication for gout, allopurinol. That's a commonly used medication to treat gout, inhibiting xanthine oxidase, the final step of the uric acid synthesis. Now, this HGPRT is a very important salvage enzyme. There is a kind of genetic disease, HGPRT deficiency, that's Lesch-Nyhan syndrome. So know the name of this, syndrome caused by HGPRT deficiency, and also know that a typical symptom or behavioral signs, self-mutilation, like chewing on fingers and lips, those self-mutilation behavior that could manifest even that person is very, very young, like a childhood. You might think that the child doing some strange thing, might grow out of it someday. It could be a sign of Lesch-Nyhan syndrome. So connect all those, yellow box things. Also, another disease, von Gierke disease. This is also linked to the elevated uric acid. That's why I kind of talked about it. Von Gierke disease is caused by the glucose 6-phosphatase deficiency. Okay, glucose 6-phosphatase deficiencies. And that deficiency results in the three things: hypoglycemia, it's got low glucose, hypoglycemia, and enlarged liver due to the lots of glycogen accumulation, the hyperuricemia, high level of uric acid. So connect those three. This is actual mechanism, but I'm not going to ask that detail. But those three items or three presentations or symptoms of von Gierke disease. And what genes are deficient in this patient? Glucose 6-phosphatase. Okay. The link between this glucose 6-phosphatase and the uric acid is through pentose phosphate pathway. So that detail you also need to know. So the glucose 6-phosphate is directly linked to the production of PRPP, which is the precursor of nucleotide production, right? So that's why it's linked to the purine production, uric acid synthesis, so on. Okay. Glucose 6-phosphate, then it accumulates going through PPP, and that will lead to the increase in PRPP, okay, pentose phosphate pathway. Direct this glucose 6-phosphate into PRPP. Or, sorry, correct. Also, there's a disease, severe combined immunodeficiency scale. That's immunological kind of deficiency problem kind of disease. Is scale. And there are several kind of genetic causes for this disease scale. But what you need to know is adenosine deaminase deficiency results in SCID, adenosine deaminase deficiency, because there's adenosine deaminase involved in the conversion of adenosine to something else. Okay. So missing this enzyme will lead to the accumulation of deoxy ATP. Okay? Adenosine deaminase will lead to the accumulation of DATP, which is the inhibitor of ribonucleotide reductase. Okay, the one I mentioned, emphasized early on. Okay, so that's the link between this enzyme to SCID. Now, basically, the inhibition of this will screw up the replication of the immune cells. So that's the kind of molecular biochemical link between immunodeficiency SCID and adenosine deaminase deficiency, ribonucleotide reductase. Right? That's it. Okay. Next one is nucleotide biosynthesis medical correlates. And about this heart pain, angina pectoris, and so the ischemia, lack of oxygen, leads to their chest pain, angina pectoris. Molecularly, the cause of this pain is accumulation of adenosine. Basically, this is a warning sign that your heart is suffering from ischemia. Okay, accumulation of adenosine. So the lack of oxygen will lead to the accumulation of adenosine. Now binds to the sensory neurons in the heart, nociceptor neurons, sensory neuron in the heart, which have adenosine receptor. So accumulation of adenosine by ischemia will stimulate the sensory neuron and send the signal to the brain to make you feel like it's painful. Yeah, so that's the kind of signal. Accumulation of adenosine binds to the adenosine receptor and the nociceptor or sensory neurons and leads to pain sensations, okay? And also remember that the action of the caffeine often included in the painkiller is the blocking of this receptor. So antagonist basically inhibiting or blocking this receptor to prevent that the adenosine from stimulating these neurons. Okay. Now going into this DNA repair stuff. Okay, DNA repair, DNA damage often occurs by various causes. The emphasis here is ultraviolet light, UV light causes damages to the DNA. I need to know what kind of damage. Specifically, so-called thymine dimer. Okay, thymine dimer. Okay, two thymine together, making cross-link or covalent kind of attachment. That's a thymine dimer, very abnormal kind of structure here. And that's going to be induced by ultraviolet light. Yeah, this is the errors in the DNA. That's going to screw up lots of things. So we constantly exposed to the sunlight, and we constantly having thymine dimer, or we have some repair mechanism to get rid of those damages. But in this patient, xeroderma pigmentosum, okay, xeroderma pigmentosum. The person is deficient in a DNA repair enzyme, so it cannot repair DNA damage caused by UV light, having all the skin problem like this. Okay, so know that, recognize the name of this disease and identify as the deficiency in the DNA repair enzyme. Cannot fix that thymine dimer, yeah, thymine dimer error, that damage is caused by UV light. Now, the rest is about methylation. So the first of all, you need to know methylation occurs on cytosine. Okay, cytosine gets methylated to make this methylcytosine. And that will affect the gene expressions and so on. And so some chemical pathway you need to know is a methylation. I just emphasized, cytosine. And this methyl group comes from the SAM, adenylyl methionine. S-adenylyl methionine is the methyl donor. And also the enzyme name, you should be able to recognize it. DNA methyl transferase, DNMT, methyl transferase. Yeah? So DMMT responsible for cytosine methylation, and methyl group come from SAM. Now we also have a demethylation process, DNA demethylation. It's a complicated process, but the first reaction is the oxidation of this methyl group. That's the first step. Oxidation of methyl group is the initial step of demethylation of DNA. And again, so no, DNMT, actually, that's it. This imprinting is interesting stuff, but I'm probably not gonna ask any questions about genomic imprinting. Vitamins, this is kind of long stuff as well, and lots of information. So pay attention. So there are lots of things I emphasize. And the basics about vitamins, but there's one question about which vitamin is a signaling molecule. So A and D are signaling molecules. Now I'll show you some slides later on. If you are asked which vitamins are acting as a signaling molecules, you have to pick either A or D, right? So A, what you need to know about vitamin A is that it's involved in vision. Vision specifically is a component of rhodopsin, okay, vitamin A derivative, specifically 11-cis-retinal. Okay, I didn't emphasize here 11-cis-retinal. You don't need to know this chemical conversion, but you need to know the name of this. 11-cis-retinal, that's made from vitamin A, is a component of rhodopsin and responsible for receiving light. Okay, 11-cis-retinal. And also vitamin A becomes retinoic acid, which act as the signaling molecules. Yeah, retinoic acid from vitamin A acting as signaling molecules. Vitamin C, make sure to connect vitamin C and the collagen hydroxylation. The hydroxylation of proline residues in collagen. So the yellow boxed those two things. Definitely need to know vitamin C essential for hydroxylation of protein in collagens. Okay? And this hydroxylation of protein is essential to make collagen fibril, triple helix. That's the foundation of this very robust connective tissue. You probably know about this, but probably there won't be a question about this. D, okay, D also signaling molecules, okay, D also signaling molecules, and signaling to control gene expression involving calcium metabolism or calcium level up. Okay, so vitamin D signaling molecule regulating calcium level. And also this is the vitamins that we can make from scratch, like a cholesterol. So that's also you need to know certain kind of vitamin D, but you don't need to know this detail. Vitamin D can be made from cholesterol using this step that require UV light, yeah, sunshine, UV light, and they'll convert this component to the components, and that's essential for this vitamin D production from cholesterol. What you need to know about vitamin E is that it acts as an antioxidant to eradicate or erase fatty acid peroxide. Okay? Vitamin E to neutralize fatty acid peroxide. Okay? Fatty acid peroxide, which is kind of dangerous, kind of reactive molecules that forms in the lipids like this. Vitamin E erase fatty acid peroxide. Vitamin K, essential for blood clotting. Okay, that is the kind of knowledge you need to know. Not this much detail. But what you need to know also about vitamin K is that there's a medication, warfarin. Okay, warfarin. It's commonly used as the anticoagulant or blood thinner for someone who excessively coagulate like this. We need a certain level of clotting to prevent excessive bleeding. But if you have too much clotting, that will block your blood flow, which is not good. So some people need to take this medication to prevent that anticoagulant or blood thinner. The action of the warfarin is to prevent the formation of vitamin K, active vitamin K. And the enzyme that's targeted by this warfarin is vitamin K epoxy reductase. Just recognize this term, the vitamin K epoxy reductase. This is an enzyme that's essential to make vitamin K active. Okay, so that when you take drug, you block this enzyme epoxy reductase, so you cannot have lots of vitamin K. Therefore, clotting is going to be reduced. And B vitamins, there are lots of B vitamins, and I will emphasize what you definitely need to know for the final exam purpose, like a vitamin B1 involved in a very important chemical reactions. But what you need to know is that medical correlations. Okay? Vitamin B1 deficiency causes this disease called the beriberi. Okay? Beriberi has a neurological cardiovascular deficit or defects. Yeah. Yeah. So beriberi, vitamin B1, berry, like the I, B1. Yeah. And okay, B2, B3 could be kind of confusing, but I don't want you to get confused. So I will just focus on B3. Okay, you can forget about B2, but just focusing on B3 in the future, will help to remember B2 as well. But for now, B3, niacin, B3, niacin. And niacin is the precursor of NAD, NADH, NADPH, so nicotine, yeah, NAD. So know that, okay? As you know, NAD, NADH, that's going to produce ATP stuff. B3, niacin, NAD, NADH. Connect those, name of this vitamin B3, niacin, and it's a precursor of NAD, NADH. Also, vitamin B5. This one, please remember the chemical name of this B5, okay? Pantothenic acid, pantothenic acid. B5 pantothenic acid, okay? And it's a precursor of coenzyme A. Yeah, there's yellow boxes that you need to know, okay? Vitamin B5, pantothenic acid, coenzyme A. Hope you're already familiar with B6, but B7, okay. B7's actions is kind of summarized as capturing CO2 for carboxylation. So in our body, there are many reactions that will add additional C carboxyl group to the molecules. And that kind of reaction often requires B7. Okay? B7 helps to capture CO2 to add the COO to the molecules. For example, there's this process in the gluconeogenesis that change pyruvate into oxaloacetate. That basically having the COO attached. Yeah? Pyruvate to oxaloacetate conversion in gluconeogenesis. That process requires B7 to capture the CO2, yeah, attached to. I emphasized this B9 importance early on, but also here I'm going to emphasize the importance of B12, B12, B12. There are only two reactions that are very important in our life that depends on B12. So just know those two. Yeah, homocysteine to methionine conversion that requires B12. The homocysteine to methionine. Also, this B12 dependent reaction. Change in methylmalonyl-CoA, methylmalonyl-CoA into succinyl-CoA. So in various kind of biochemical process, our body creates this methylmalonyl-CoA. And this is a kind of not important, or actually you don't want to accumulate those things too much. But this enzyme will change this into succinyl-CoA. That enzyme depends on B12. Yeah. So that means if you don't have B12, this reaction is going to stop. You're going to accumulate methylmalonyl-CoA, something like that. Yeah. B12, adding one carbon, homocysteine to methionine, methylmalonyl-CoA to succinyl-CoA. Both are B12 dependent. That's it. Okay. Clinical biomarkers. And biomarkers, what you need to know is no specific biomarkers for specific disease conditions. Myocardial infarction. There are two biomarkers that are commonly used. Creatine kinase, the creatine kinase, or CK, creatine kinase, is one of these myocardial infarction biomarkers. And there are CK, there are different kind of creatine kinase in the same function, like the same enzymatic actions, but it's a different molecules in the sense that amino acid sequence is different, so that way it's different. But it's responsible for the same enzyme reaction. That's why it's isozymes. And there are different isozymes, CK1, 2, 3. But CK2 is specifically made by cardiac muscle. Okay, that can be distinguished by some method you don't need to know. CK2, one of the isozymes, CK isozyme, is produced specifically cardiac muscles. So that's why the release of CK, this CK2, can serve as the biomarkers for cardiac muscle damage, destructions. Yeah, damage, release, show up in your blood, can be detected. And the second biomarkers for myocardial infarction is troponin. Okay, there are specific troponin molecules present only in the cardiac muscle. Okay, so this one. But what you need to know here is what is troponin? You know, troponin is kind of general normal function, which is to regulate interactions between thin and thick filament. Yeah, for muscle to contract, you have to slide this thin and thick filament against each other. Yeah, that sliding or interaction, that's regulated by this troponin. And pancreatitis, there are biomarkers. Basically, what you need to know is two enzymes, amylase and lipase. Amylase, lipase, both are responsible for digestion of food, yeah, like here. But amylase, lipase serve as a biomarkers for pancreatitis. And also liver damage. What are the biomarkers of liver damage? And like elevated ammonia that can serve as the accumulation ammonia is the indication of liver function failure. So the biomarker of liver damage. And also the jaundice. Yeah, liver is responsible for changing bilirubin to soluble form. So lack of this, or the deficiency or incompetent liver, damaged liver, end up accumulation of bilirubin, therefore jaundice. Got it? Okay. All right, two more to go. Drug development number one. So kind of had examples of diabetes medications. So you need to know specific category of diabetes medications. First one is the inhibitors of ATP sensitive K plus channel. Yeah, you don't need to know the names of this actual medications, but what you need to know is you're basically targeting this beta cells. This drug target beta cells. This cascade of event that normally happens in the beta cells to release insulin, right? And one step here is the inhibition of K plus channel. That's normally should happen in response to high glucose to release insulin. So this drug is basically mimicking the action, inhibiting this K plus channel. So regardless of this upstream events, this drug will directly inhibit this, therefore causing the next step, which is promoting insulin secretions. So that's the action of inhibitor of ATP-sensitive K plus channel. And another category of diabetes medication, inhibitor of DPP4. In this context, it's a bit complicated, but what you need to know is that the very important peptide hormone here, GLP-1, you heard about one of the research groups talked about Ozempic, right? Ozempic is kind of imitating this GLP actions to promote insulin release. GLP-1, this is a normal hormones from the intestine that will act on beta cell to promote insulin. So you probably need to know this part very well. And this medication is acting indirectly on this one by blocking this DPP, which is to destroy GLP-1. But I want you to actually know more about this GLP as a main guy, okay? Coming from the intestine and acting on the beta cells, promoting beta cells to release more insulin, GLP-1. And the third drug is alpha glucosidase inhibitors. Yeah, it's basically preventing the digestion of this carbohydrate and to reduce glucose absorption. Yeah, and they're blocking the split of this maltose into glucose. So less glucose in the intestine, so reduce glucose absorptions. And the fourth one is inhibitors of SGLT2, sodium glucose cotransporter 2. What you need to know is that this drug is acting on kidneys, okay, kidneys, because kidney is the place that filter glucose and reabsorbing this glucose. So the kidney is doing some complex stuff here, but this drug is preventing this reabsorption of glucose, basically promoting glucose excretion. That's how you try to reduce glucose in the body. Yeah, SGLT2 inhibitor acting on the kidney to promote the glucose excretion. Those yellow boxes, those are the important main parts that you have to focus on. Okay, so the ADME stuff, there's certain thing I want you to know for the exam, which is the route of administration, oral pathway. Okay. This is very, very important because you take oral drugs quite often. So you have to understand that what kind of journey this oral drug has to go through to reach the entire body circulation. So there's 5 steps here, right? Oral to stomach, intestine, portal vein, which is this blue blood vessels from intestine up to liver. Yeah. And then liver up here into the heart, and heart's going to pump out those drugs into the entire body. You need to know the order of this 5 steps. There will be question. I promise you, there will be a question. You have to find out. There's a 5 steps here. Which one is number 3, number 4, number 5, that kind of questions. So you have to be able to shuffle those 5 elements in a correct order. Please. And D, ADM is D, distribution, various things affect how drug can distribute in our body. And a good example of this distribution is the blood-brain barrier. We're going to talk about this before in the different lectures. Some chemical cannot pass through this particular layer between blood and brain, blood-brain barrier. So those are going to affect the distribution. Okay, blood-brain barrier affect drug distribution. Okay, the example of distribution like a blood-brain barrier. Metabolism, okay, you need to know some aspect of metabolism specifics. First of all, it happens in the liver. One of the research group presented about metabolism. We emphasized the CYP enzyme, yeah. Metabolism, liver, and one of the enzyme is cytochrome P450 or CYP enzyme. There are different kinds of CYP, yeah. And that's most of them are responsible for oxidation, attaching O to make hydroxy group, oxidation. CYP, cytochrome enzyme, oxidation in the liver. Just to be aware, as a kind of general, like a concept, there's certain things can interact with other drugs and the foods, affecting like a metabolism or distribution, that sort of thing. So if you take drugs, with some other drugs or food that would definitely affect that activity or distribution or metabolism of the drug you're taking with. Yeah. So sometimes that there's a label or the doctors instruct that don't take this drug with certain food or certain other drugs kind of thing. So there's interactions through this metabolism or distribution. That's the conceptual thing that you need to know. Okay. All right. One more thing. Okay. So the toxicity part of this drug development. Okay. So the flow of drug development, this is, probably, I would say that the most countries follow this kind of flow, but this is the sort of United States way, uh, Food and Drug Administration. So made of this, first need to know this flow, preclinical drug discovery and 1 to 3 phases of clinical trial, human clinical trial, and then clinical use, like a market it, and the doctors start to prescribe to the regular people and so on. You need to know the flow, okay? And what also you need to know is that preclinical phase basically is done using in vitro animal models to study drug efficiency or drug toxicity. Yeah, preclinical discovery phase involve animals or in vitro system, not humans at all. Okay, human trial is approved or permitted only after extensive preclinical studies. And historically, the thalidomide is important drugs because this is the first drugs that are identified as something outside can affect the developing embryo. Yeah. So this was widely available or used, prescribed for pregnant women for morning sickness treatments. It was very, very good for women, but it caused the birth defect to the developing embryo's fetus. So that therefore it was withdrawn from the market. Thalidomide, okay? Recognize the name of this drug, thalidomide, causing birth defects. Also, just be aware of this term, teratogens. What is teratogen? Chemicals that cause embryo death or birth defect. Teratogens are chemicals that cause embryo death or birth defect. And those embryos are affected by those teratogen, various those chemicals, disturbances. And embryo development is very dynamic, especially dynamic during the early phase, like day 0 to week up to 8. Lots of things happening, dynamically changing from the single cell to complex bodies like this. Yeah. So those early stages of pregnancy are particularly vulnerable to chemical insults like teratogens, particularly sensitive. So this is a period that women need to be extra careful. Yeah. So that's the phases. They are first to 8th week, those are very, very sensitive because it's dynamically changing, easily affected. So the FDA categorizes drugs based on the knowledge, their research data and so on, safety for the pregnant women, pregnancy risk, A, B, C, D, X. I want you to know the definition of category X. Just read this sentence 3 times to understand what category X means. Okay, read the sentences 3 times to know what category X drugs mean. And actually, yeah, it seems like I'm not going to ask anything about embryonic stem cells, my research at all. It's not like my research not important, but I'm not going to ask anything in final exam. Yep, that's it. All right. So again, so this the study guide is the 70 multiple choice questions of the final exam. There are additional 15 questions from your the classmates made. I will send out a separate email, okay, which is gonna hint about what kind of questions, this research project-based questions, like what to focus on, what to emphasize. I'll give you some idea of some hints. Right now, ideally, since those other groups, the PowerPoints are uploaded to Launima by then, ideally, it's good to sort of go through again just to know what they discovered or studied or compiled information for us, for you guys. So, but I will help you with some hints that what specifically you want to focus on. And I will send out that email probably after the all the presentations done on Wednesday, May 1st, but maybe a day later or a couple of days later, as soon as possible. Yeah, after I select which one, and I'll emphasize what aspect to focus on. All right. So one more thing that lastly, if you haven't done so, then please fill out the surveys for our course and through this CES here. You probably received some email asking you to fill out. And this is kind of important for us, for the instructors. And so that Dr. Ward, Dr. Holger, and myself, I will definitely really appreciate you if you fill out. Because we want to know your opinions. How did you think about this course? What you liked about it or what you didn't like about it. I personally want to hear about what you didn't like kind of stuff too, because that help us to modify, improve this course for next year. Yeah. So some positive, some critical feedback. We really appreciate it, your honest opinion. Yeah, it's totally anonymous. So no matter what nasty thing you say, I mean, you can be negative, that's fine, but let's be a little more professional. Don't be too emotional kind of thing. But if you find something is not good, you can tell us how we should change or improve for the future. So I want to hear. We definitely want to hear about good thing, what you liked about this course, so we can preserve those good things for the future as well. So I want to hear both aspects. Yeah, I would really, really appreciate if you can fill out. I know it's a time consuming. I don't know how long, but I know you're busy, but I would definitely really, really be grateful if you can spend some time to fill out this survey. Okay. Other than that, if you have any questions about what I talked about in this video, please email me. I'll respond to you as quickly as possible within 24 hours, I guarantee. So yeah, any questions? Let me know. All right. So with that, I'm gonna end this study guide. All right. Good luck.